This lecture is about cell biology, and it's just enough cell biology to familiarize you with some of the terms that we're using in the rest of this course. We're not trying to give you a detailed background in cell biology, which is a very dense and complex topic, and you could spend years studying it. In fact, people spend their whole career studying it. But cell biology and molecular biology include many complicated sounding terms, and it's important that you understand what these words mean because we're going to be using them throughout the course. So at the most basic level, we divide cellular organisms into three domains: eukaryotes, archaea, and bacteria. And archaea and bacteria are sometimes grouped together and called prokaryotes. And the distinction we make between these two very fundamental groups of living things is that eukaryotes have cell nuclei and prokaryotes do not. Now, evolutionarily, these three domains of life split off from one another very, very long ago, near the beginning of the evolution of life on Earth. Bacteria and archaea split off a little bit later after eukaryotes had split off from one another, and bacteria and archaea separated. But bacteria and archaea, in many ways, at the cellular description level, seem very similar. The big difference between eukaryotes and prokaryotes is that prokaryotes do not have a cell nucleus, and eukaryotes do. And the cell nucleus evolved as a long, long time ago as a way to sequester our DNA from the rest of the cell. So at the cellular level, eukaryotic cells and prokaryotic cells look rather different. The biggest difference is that a eukaryotic cell has a nucleus. So even single-celled eukaryotes like yeast, which is a very common single-celled eukaryote, you all probably eat it every day if you have bread. Even yeast have nuclei just like our cells do. So humans, yeast, and everything in between are eukaryotes. Our cells have nuclei in them. And other organelles or small cellular structures as well. Prokaryotes don't have that. So in particular, the DNA in a prokaryote is, you can think of it as just sort of floating around. It's not really floating, but you can think of the DNA in a prokaryotic cell as just kind of loosely organized inside the cell. And in eukaryotes, there's a bit more organization. In eukaryotic cells, DNA is sequestered deep inside. So if you look at this picture here, inside the eukaryotic cell, which is surrounded by a wall, there's a nucleus which has its own membrane or wall surrounding it. And inside that nucleus is where we put our DNA. And our DNA is organized, of course, into chromosomes, which are these very, very long molecules of DNA. And all your chromosomes are inside each cell, and within those cells, they're inside the nucleus in each cell. And there's one slight exception about your DNA, which is that one of the organelles inside of a eukaryotic cell is called the mitochondrion. The mitochondrion, and actually there are multiple mitochondria in each cell, so the mitochondrion has its own DNA. In the human genome, the mitochondrial DNA is very, very small. It's a tiny fraction of 1% of all of our DNA. But you might have heard mitochondria called the powerhouses of the cell because the genes inside your mitochondria are responsible for a good bit of energy metabolism. So even though it's very small, doesn't have that many genes, it's absolutely critical to life. And as an interesting sort of side note, we believe that the mitochondrion, very long time ago, billions of years ago, was originally an independent prokaryote that was absorbed by the ancestor of all eukaryotic cells into the cell itself, and then has become part of every eukaryote now on the planet. So all eukaryotes have mitochondria, and they've diverged since the various eukaryotic species started to diverge. And most of them are very small. Back to the cell itself. So cells undergo a cycle, a characteristic cell cycle that many people study. It's not important that you know all the names of the parts of this cycle, like metaphase and anaphase. But cells, during the course of their life, they have a fairly well-defined cycle where they undergo different processes. One of which, the most critical, is the process of division. So the way you go from a single-celled progenitor cell, from a fertilized egg to a whole organism, is the cell has to divide many times. And throughout your life, your cells are constantly dying and have to be replaced. In order to replace damaged tissue, your body has to divide, which means cells have to divide to replace the cells that are damaged. So that part of the process is called mitosis, where a cell separates into two daughter cells, producing two essentially identical cells. And for the purposes of our discussion, it's important that you realize that you understand just a couple of words here. So when a cell undergoes mitosis, the DNA in that cell has to replicate. That is, it has to make two copies where there was one before. So inside the cell, before it divides into two copies, the DNA replicates. Now you have two copies of every chromosome, where there was you actually start off with two copies of every chromosome. After replication, you have four copies. And now the cell has to figure out, through a complicated procedure during mitosis, how to separate those copies. And that's what happens. The copies that are made are actually separated very reliably into different physical compartments in the cell as it's dividing, and as a result you get two daughter cells, or as we call them, that are identical to the original cell. And both the daughter cells are also diploid. So there's another word I haven't used yet, diploid. So not all eukaryotes are diploid, but we are. And diploid means that we have two copies of every chromosome. So our DNA exists in two copies, one copy from mom and one copy from dad. And of course, in diploid organisms, it's important that every time a cell divides, it maintains these same two copies of every chromosome. Another important aspect of cell biology is that cells don't always divide and produce two identical copies. Of course, if you're replacing existing cells that have been damaged, say you've got a cut in your skin and new skin cells have to be grown, you want the skin cells to be just like the ones that were there before. But during the course of development, cells need to develop into different types of cells. This is a much more complicated process than we have time to go through, but all the cell types in our body start as more basic cells that we call stem cells that have the capability of dividing and differentiating into different types of cells. So this process of cell differentiation involves a slightly different cell division process where the two daughter cells are not identical to each other, and they go down what we call developmental paths that allow them to differentiate into mature cells of different types. So all the cells, for example, as this slide is showing, all the cells in your blood start as what we call a multipotent hemopoietic stem cell, which can divide and produce many different types of blood cells. And the cells circulating in your blood all came from these stem cells. And the stem cell populations in your body are maintained over time as well. So there's a special type of cell division I also want to mention in this brief introduction, and that's called meiosis. So during sexual reproduction, something different happens in cell division, and that's the main point we want to get across here. When you're producing egg cells or sperm cells for the next generation of your species, you start off with copies of chromosomes from the mother and the father, and those are all put together before meiosis in a single compartment. And then there's a special process that happens. It's not by design, it just happens by accident, called recombination, where the chromosomes, because they're very, very similar, they can kind of stick to each other, and we have this process called crossing over that occurs, where part of say, chromosome 1 from mom can cross over with part of chromosome 1 from dad, and you get resulting in the daughter cell. You can have a copy of chromosome 1, which is partly mom and partly dad. Now, these recombination events are relatively rare, but not that rare. And in humans, in fact, we estimate that in each generation, there's approximately one crossover event per chromosome. So most of your chromosomes, although you got every bit of your chromosomal DNA from your parents, your actual chromosomes end to end are not likely to be identical to either of your parents' chromosomes. So recombination is one of the big reasons that besides mutation, is one of the other reasons why family members aren't alike. So I'll just conclude by saying that during the course of cell division to produce children, every child has different recombinations. So even though every child of the same set of parents has essentially the same DNA as the two parents, there are an almost infinite number of possible recombinations, and not quite infinite, but a huge number of recombinations that occur. And so every child actually has a different set of chromosomes, although you can clearly trace them back to both parents. And that's one of the big sources of diversity in our population.